Hello everyone, and welcome to today's webinar, Advanced Enzymes to Facilitate SARS-CoV-2 Research. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting as many questions that you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Lavelle, Senior Product Manager, Reverse Transcription at Thermo Fisher Scientific. Lee, you may now begin your presentation.
Thanks, Jennifer, for the intro. Hello, everyone. I appreciate you attend this webinar, and it's my pleasure to share with you the information about how next-generation engineered enzymes were used in the SARS-CoV-2 research. Let's go ahead and start. Due to COVID pandemic, a broad range of interests have been triggered in understanding the mechanism of the virus and identifying solutions to address this global crisis. A lot of efforts goes into the viral research, virus detection, prevention, and treatment. For the viral research, there are studies focusing on genome transcriptome characterization, pathogenesis mechanism, virus evolution, and other aspects. The detection of SARS-CoV-2 has become the focus of research programs. There are also a number of initiatives aiming for vaccine and antivirus therapeutic development. Now we come to our very first poll question. I was thankful that you will take a little bit time to fill out this poll question. Okay, let's continue. Currently, SARS-CoV-2 virus research is focused on three directions according to the technologies used. Their target detection, high-throughput sequencing, and cloning. For target detection, RT-qPCR is the gold standard, has been adopted by majority labs for virus detection. The researchers also explore RT-LAM and the CRISPR-based methods for point-of-care use. I'm going to share a little bit more information about target detection in the next few slides. For high-throughput sequencing, NGS is the dominant method with flexible strategies. For example, the researchers can choose to use whole genome or targeted sequencing or whole transcriptome analysis. Other than that, single molecule long-read sequencing, such as nanopore sequencing, also gained popularity in virus research. I will share more in the later slides. Cloning has been used in many applications that involve RT-PCR, Sanger sequencing, a number of cloning techniques. The current COVID-19 pandemic presents a serious public health crisis, and a better understanding of the scope and spread of virus will be aided by more widespread testing. Here's three different types of nucleic acid testing has been explored. So I will use this slide to give you an overview. The standard RT-qPCR method is being widely adopted by a lot of labs. Patient nasal swab or saliva samples are collected and the viral particles are inactivated and lysed, cDNA synthesis and subsequent PCR amplification are performed with either a crude sample or purified RNA targeting specific regions of the virus genome for detection. A popular alternative to RT-qPCR is isothermal amplification, which does not require thermal cycling. The major techniques are loop-mediated isothermal amplification (LAM). I'm sure you have heard about this term before, and the recombinant polymerase amplification (RPA). LAM uses a strand displacing DNA polymerase with specifically designed nested primers with complementary regions that form hairpins to permit priming of subsequent rounds of amplification. The same process repeats, and repeated rounds of priming and strand extension generate a mixture of stem loop and cauliflower structured products. The release of hydrogen ions can be detected using colorimetric pH indicator dyes. The RPA uses a recombinase to catalyze strand invasion of primer into double-stranded DNA. Single-stranded binding proteins are included to stabilize the open duplex structure, and strand-displacing DNA polymerase extends the primer. Some researchers use both RPA and the LAMs in a two-stage amplification protocol to improve specificity and sensitivity. The third category is CRISPR-based detection method. A unique group of Cas nucleus, such as Cas12 and Cas13, were discovered to possess DNA or RNA cleavage activities. This feature has been exploited for nucleic acid detection. The Cas non-specific nucleus remains inactive until it binds its programmed RNA or DNA target. The reporter molecules are cleaved, releasing a fluorescent dye from a quencher. The right-hand graph shows the limit of detection across different methods. You can see RT-qPCR is still the most sensitive method. That's why the majority testing still come from the RT-qPCR. Other methods can use to detect as low as 10 molecules. Okay, let's take a look at the second poll question. This slide shows you SARS-CoV-2 analysis workflow. It includes RNA extraction from a variety of sample sources as listed here. Nasal swab and saliva samples are the more common SARS-CoV-2 samples. And researchers can pick environmental samples, blood samples, and other types of samples to extract RNA. The purified RNA will be reverse transcribed and amplified, then analyzed in different ways based on the protocol workflows. Some examples are electrophoresis, real-time fluorescence, or sequencing. SARS-CoV-2, what I want to point out is in this slide is sample preparation and enzyme choice are interconnected. Usually, a good high-quality RNA sample may only require regular enzyme like MLV, reverse transcriptase, or Taq DNA polymerase. However, if degraded RNA samples or low-input RNA is used, a better enzyme is required in this case because the better enzymes help deal RNA or DNA targets of suboptimal purity and abundance. SARS-CoV-2 has emerged in 2019 and has rapidly spread worldwide. It is a positive sense single-stranded RNA virus with 5 cap and 3 poly A tail. It's the longest RNA virus, about 30 kilobases. The clinical viral samples are easily degraded during transportation and RNA extraction. Other challenges include long testing time and multiplexing challenges. A number of features should take consideration when you select enzyme reagents to study SARS-CoV-2 virus. The preferable feature of the enzyme include high processivity, fast speed, better specificity, accuracy, and ability to multiplex should be considered for SARS-CoV-2 research. Here, I just want to point out processivity is defined as the number of nucleotides incorporated during a binding event. For example, the wild-type MLV reverse transcriptase can incorporate 50 nucleotides in one binding event. With engineered next-generation enzymes such as SuperScript 4, the incorporation number much higher. It could accomplish 1,500 nucleotides incorporation in one binding event. So the processivity is much higher. And this can be translated into the high speed. So some next-generation enzyme examples listed here. For the reverse transcriptase, SuperScript 4 reagents, they're all pretty good candidate for study to SARS-CoV-2 research. And for the DNA polymerase, Plantium Superfy 2 DNA polymerase currently has the highest fidelity, is a 300-fold more fidelity than any current high-fidelity DNA polymerase on the market. High fidelity is very important, essential feature to be used for cloning applications and sequencing applications. Okay, now we can zoom in to talk a little bit more about viral target amplification. I'm going to share two methods, RT-qPCR and RT-LEN. And the featured reagents are listed here. It's SuperScript 3 one-step, and SuperScript 4, also BST DNA polymerase. I'm going to share a number of publications. WHO recommend a number of RT-qPCR protocols for SARS-CoV-2 detection. This protocol using SuperScript 3 and Plantium tag is one of them. Following the recommendation, a German group tested this protocol and purified RNA samples amplified using one-step RT-qPCR with gene-specific primers targeting RD-RT and E genes. The unspecific signals were significantly reduced in the SuperScript 3 one-step setup compared to QuantiTag, another RT enzyme, which used as a reference. This protocol is demonstrated to be a fast and sensitive protocol for virus detection. The protocol can be downloaded on WHO website. I listed a link on the bottom of this slide for your reference. In an effort to block the widespread transmission of SARS-CoV-2 virus, researchers from Georgia State University tested the efficacy of drug compound MK4482-EIDD2801 against SARS-CoV-2 infection in ferret models. This compound has shown potent anti-influenza virus activity in the clinical trials. Viral administration of the drug compound exhibited reduction of virus load in the upper respiratory tract and successfully blocked community transmission. RT-qPCR was used to detect the virus load with RNA purified from either harvest organs or PBMC samples. In addition, The systematic interferon and cytokine expression profiling is performed with real-time cyberbrain and assay. Now, I would like to spend a little bit of time to compare the differences between the gold standard detection method RT-qPCR and RT-LAM. RT-qPCR method requires trained personnel and a fully equipped lab facility. RT-LAM is a more simplified method, easy to use, no special equipment needed. It takes shorter time to generate results compared to RT-qPCR. However, RT-LAM has lower sensitivity and specificity than RT-qPCR. Recently, we published an application note detailing an optimized RT-LAM protocol. This slide shows the simplified RT-LAM protocol workflow designed by us for rapid detection of SARS-CoV-2. This protocol does not require new reagents. All the reagents used in this protocol are existing reagents, can be ordered on our website. It also provides template to test the protocol for additional targets with other primer sets. The protocol starts with Magmax purified RNA samples. All the components are combined together to prepare reaction mix. The reaction mix include BSM, DNA polymerase, SuperScript 4, reverse transcriptase, RNA template, and specifically designed 6 primers. After mixing all the components together, and the protocol goes through the incubation and inactivation steps. Finally, several endpoint methods to visualize the results are demonstrated, such as direct color visualization shown on the picture A, and agar gel shown on picture B. So you can see the SARS-CoV-2 positive samples and shows that different target band from the lane 1 to lane 5. And the Korean, there's a Korean group, and they actually looking at develop their own version of LAM, RT-LAM. The design evaluation RT-LAM assays for detection of SARS-CoV-2 with limited of detection of 100 copies per reaction. So they actually extract the RNA from a culture medium of the SARS-CoV-2 infected cells, and SuperScript 4 reverse transcriptase with cDNA polymerase and used for cDNA synthesis and amplification. And the visual colorimetric visualization can be performed. As shown in the graph on the right-hand side, you can see is this method sensitive enough can only detect color change with SARS-CoV-2. In no template control and some similar virus sequences, and you don't see the color change. So the visualization, the detection can be also performed with RT qPCR method. Now let's take a look at the deep sequencing for virus detection research. We switch gear to take a look at the NGS method and nanopore sequencing and other sequencing methods. So SuperScript 4 reverse transcriptase typically used for this type of applications. We are going to look at a number of applications. Deep sequencing plays a critical role in SARS-CoV-2 research. It offers single-base sensitivity to investigate virus variants. There's no prior sequence knowledge required compared to PCR-based methods. It's also a common method to study host pathogen interactions with complete and high coverage. Researchers can choose whole genome sequencing or target sequencing NGS, or single molecule sequencing. The scales of the studies can be large or small. Australia researchers analyzed COVID-2 transmission pathway in Australia by SARS-CoV genome sequencing. This publication happened a few months after the global pandemic happened. So they want to identify which portion actually locally acquired, which portion of cases actually imported from overseas. They collected clinical samples, the clinical samples with positive RT-PCR results. These samples were reverse transcribed using SuperScript 4 Velo master mix. The viral cDNAs were amplified by multiplex PCR with Plantium Super5 PCR master mix. Amplicons were then pulled, purified, and quantified, followed by library prep using XT library preparation kit. Only a quarter of the sequenced cases appear to be locally acquired. This high-resolution genomic data are crucial to track cases with locally acquired COVID-19 and overseas importation. How do we decide if enrichment is needed for sequencing? So this really depends on research goal. If the purpose is just focused on the viral genome, the viral genome needs to be enriched by either amplification based on hybridization or amplification method. The overall sequencing cost will be reduced. The majority reads would map to the SARS-CoV-2 genome. However, if the research goal is to analyze host pathogen interactions, no enrichment will be required. You will gain a whole picture with reads covering a variety of different genomes, with human genome over 70% and less than 3% of reads mapped to SARS-CoV-2 genome. We developed an optimized workflow for sequencing the SARS-CoV-2 genome. You may download it from our website. Download the app notes from our website. This application note details the protocol starting from RNA purification. Total RNA was extracted from BAL clinical specimen. The applied biosystem MagMax viral nucleic acid isolation kit was used for purification. SuperScript 4 Velo master mix and Thermo Scientific second-strand cDNA synthesis kit was used for cDNA synthesis. The sequencing ready library was prepared with Calibri Enzymatic sharing DNA library preparation kit with unique dual indexes. The library were pulled and enriched using a synthetic probe panel by PCR amplification. This step is optional. The final concentration of the libraries were quantified with Calibri library quantification kit, then sequenced on the Illumina system. Here lists the time takes for each step, so the entire workflow may take 1 to 2 days. So this is a nice study published on Cell, and last year, the Korean researchers, they established a high-resolution SARS-CoV-2 transcriptome map, and they actually used two sequencing methods to do the studies, and one is the NGS sequencing by synthesis, like Illumina or MGI system, and they used the nanopore-based direct RNA sequencing for the rest of the studies. So they started total RNA actually purified from the virus-infected cells. And the viral cDNA synthesis using SuperScript 4 RT enzyme followed by the PCR amplification and gel purification. And then they will perform the in vitro transcriptome, and they were able to follow the manufacturing protocol to perform direct RNA sequencing library and using SuperScript 4 RT enzyme. This is a two-arm approach using two different sequencing systems to verify the data. So what they found is there are numerous discontinuous transcriptome events. At least 41 potential RNA modification sites were identified. SARS-CoV-2 also produces RNA encoding unknown open reading frames. First, so this is a really nice study to map out the entire transcriptome map. So this study showing the recently we see that there's a new variant strain and called B117 variant, and the first identified UK Kingdom, and everyone knows about this strain, is really have a high profile on the medium. Everybody report about it, and it had gained a strong foothold around the world. And in December in California, first, and the researchers identified this the virus, and they collected a number of clinical samples, and those samples already verified with RT-qPCR as positive samples, and from there they will, and using both RNA sequencing and RT-qPCR two methods to confirm the B117 variant. And they show the B117 rapidly become the dominant SARS-CoV-2 variant in the U.S. U.S. will share the similar trajectory as other country where the new variant B117 is spread. So this study shows another way to analyze the SARS-CoV-2 with NGS. So currently, when the patients show the symptom, they get detected to show a positive and negative. So the early detection using RT-qPCR is the only way to predict viral load, even though there are some patients with no symptoms. This is the imperfect readout in this case. So a group of researchers from National University of Singapore, they determined to identify a better predictor of the viral load. They developed a viral long fragment score. This is based on the sequencing of the viral long fragment. First of all, DNA RNA purified from nasal saliva samples, and the reverse transcription performed using applied biosystem high-capacity DNA reverse transcriptase and amplified by the multiplexing PCR using Plantium Superfy 2 DNA polymerase. The library is prepared following the regular protocol, and then the Illumina system was used, and paradigm 2 by 150 base pair sequencing was performed. And that they proved that NGS actually indeed a very sensitive method to detect long RNA fragments. The abundant long fragment and the score is positively associated with viral load, and inversely associated with the time from the disease onset. In other words, the longer the disease has been diagnosed, the score, the viral long fragment score will be lower. So they designed the primers for 327 amplicons and with 155 SARS-CoV-2 positive samples. Okay, so after sharing two technology, let's switch gear to take a look at cloning. So cloning typically involved RT-PCR, a different cloning technique, and single sequencing. So the popular product used for the cloning applications includes SuperScript for one-step and first-strand synthesis systems. Plantium Superfy 2 DNA polymerase, another popular option because of its high fidelity. Okay, here's our posed question. I appreciate you answered that question. Let's wait here for a little bit. Now we move to another publication. A German, a group from the University of Texas, they actually developed this method called ISA, and this is they established reverse genetic of coronavirus by infectious subgenomic amplicons method. So this method has been developed in recent years, focused on the short single-strand RNA genome. And this is the first publications they established on the first publications established on the long RNA strand. So the researchers, and they use this technology and with a purpose known in need for constructing a full genomic cDNA mutations, and fluorescent reporter genes were incorporated in this case. So first of all, they designed 8 overlapping de novo synthetic viral fragments. They use this fragment as a template to synthesize cDNA using SuperScript 4 one-step RT-PCR system. Then the cDNAs were amplified with Superfy 2 DNA polymerase. And this amplified fragments were transfected into cells. The full-length recombinant viral genome was produced within the cells through the in vivo DNA combination. The full-length viral genome was then verified by one-step RT-PCR assay. The constructed genome, it turned out, is completely identical to that of targeted virus. The rescued virus replicates the phenotypic and genotypic characteristic of the regional isolate. University researchers developed another reverse genetic system to study SARS-CoV-2 virus. The viral RNA purified from viral E6 cells were used as a template for RT-PCR to produce cDNA fragments. There are 7 cDNA fragments were constructed to overlap the entire viral genome. So the 4 of them are chemically synthesized. 3 of them went through the traditional cloning process because those 3 fragments not stable by the chemical synthesis. So the cDNA was synthesized using SuperScript 4 first-strand cDNA synthesis kit, and then amplified using Superfy 2 polymerase. All cDNA fragments are individually cloned. The RNA transcribed from the full-length cDNAs were highly infectious, exhibited similar plague morphology, viral RNA profile, and replication kinetics. And in addition, the system generates stable reporter genes and can be used to evaluate the antivirus activities of potential therapeutics. So it turned out to be a really valuable system for the drug screening process. A German group analyzed antibody response of 12 COVID-19 patients from 8 to 69 days after diagnosis. The neutralizing antibody showed a broad spectrum of variable genes and a low-level somatic mutations. This demonstrates SARS-CoV-2 neutralizing antibodies are readily generated from a diverse pool of precursors and giving us the hope for rapid induction of a protective immune response upon vaccination. In this study, SARS-CoV-2 active domain-specific B cells were isolated from PBMCs. Single-cell amplification antibody having light chain was performed using SuperScript for reverse transcription, planting tag hostile polymerase for amplification. The PCR products were then analyzed by gel electrophoresis followed by Sanger sequencing. So lastly, and I would like to share with you about the benefits of Lyo-ready enzyme formulation. It requires in a process called lyophilization. This is a water removal process used to preserve perishable materials to extend shelf life or make the material more convenient for transport. Drug and diagnostic assay developers, they're interested in this process. However, the conventional formulation enzyme contains up to 50% glycerol, which stabilizes enzymes but interferes with lyophilization process. A good Lyo-ready enzyme should be formulated with glycerol content level well below 0.5%. In the meantime, maintain equivalent performance of reproducibility, sensitivity, and specificity to the compared to the conventional formulation. Okay, let's pause here for our last poll questions. So when considering a Lyo-ready enzyme for nucleic acid amplification, there are 3 different categories you can consider. One is performance. Here lists the different performance important is inhibitor tolerance, thermal stability, processivity, specificity, low residual DNA contamination. And in terms of consistency, you would like to pick the Lyo-ready enzyme and have very similar good QC tests from batch to batch and minimize any variation introduced from the different batches. And the quality will be good to pick from the facility, ISO certified facility, high quality facility to manufacturing Lyo-ready enzymes. Okay, this concludes my presentation today. I appreciate any questions you may bring up. Thank you. Okay, here is our last question, last poll question. Thank you for your attention.
Thank you, Lee, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, what's the multiplex capacity of the platinum superfy DNA polymerase? And how does universal annealing temperature work?
That's a very good question. Actually, there's no limitation with the enzyme itself in terms of how many multiplex reactions can be incorporated. It can be used for many, many targets in one reaction. The limitation factor actually from the different components. It really is a combination of how much primers in one reaction and what's the other component. That's the limiting factors. The more in reaction put together in one reaction, the more enzyme, the more primers, and the more buffer, everything has to be optimized. But the enzyme itself can accommodate as many reactions as possible. And in terms of universal temperature, that's a really, really good feature we have with Superfy to polymerase as well as Plantium Taq hard start enzyme. So we have this innovation to optimize the buffer condition. So in the past, it's very inconvenient when designing the different primer sets and have to optimize the melting temperature to a similar level. So make it the annealing temperature is more or less the same. But with the current formulation, and you can design the whatever primers you needed without worrying about the annealing temperature. You can just throw all your primer sets together, and then the buffer condition will take care of that. They will be able to use the universal condition to carry out the reaction. Hopefully I answer your question.
Thank you, Lee. Our next question here is, for detection of the COVID virus, what's your thought on using purified RNA versus adding samples directly to the RT reaction?
That's another good question. Initially, when the research developed the detection method, workflow always started with purified RNAs. There's some limiting factors. With the global pandemics, the more and more need for widespread nucleic acid testing, there's not enough the RNA purification kit. That's become bottleneck. I see that there's a significant slowdown in the diagnosis process because the limitation from the RNA library preparation kit. And so there's more and more researchers think about alternative solution, which is how about we skip RNA purification, just lyse the samples and directly carry it out to the RT-qPCR or RT-LAMP. This is a good concept which is being studied a lot. The con for this method actually is the compatibility between the transportation medium. As you know, those clinical samples, saliva or the nasal swab, need to put in the transportation medium, transfer to the lab, and then process for the next step. If the transported medium not compatible with SuperScript or reverse transcriptase in general, and that's create a problem. And there's some considerations like sensitivities and different background noise also need to consider. But in general, in direct approach, using crude samples will be faster, more straightforward, and can bypass that and bottleneck from the RNA isolation kit supply issues.
Okay, great. Thank you so much, Lee. And our next question here is, can you please elaborate more on processivity?
Yeah, processivity is, as I mentioned in the presentation, is defined as the number of nucleotides cooperated in a single binding event. So the wild type typically have 30 to 50 nucleotides incorporated in one binding event. You can imagine when they incorporate a low number of nucleotides, it takes longer time to finish the cDNA synthesis, and using reverse transcriptase as an example. And for the engineered enzyme, a SuperScript 4 is much faster. It can incorporate as many nucleotides as possible. So far, we see that 1,500 nucleotides, and there's a 500 fold difference basically between the wild type and the engineered enzyme. So the advantage of this, as I mentioned earlier, is fast, right? It's just moving really fast. It takes only 10 minutes reverse transcription compared to the wild type RT enzyme. And also it can translate to how we can overcome the RT inhibitors in the reaction mix. Typically with a lower processivity enzyme, when there's some inherited or introduced the inhibitors in the reaction, the RT enzyme the traditional RT enzyme will slow down and will not be able to finish the synthesis of full-length cDNA. But with high processivity enzyme, it will be overcome any inhibitors or suboptimal RNA qualities, be overcome that issue, still get pretty decent cDNA synthesis results. Yeah, that's a really big benefit for the next generation enzyme.
Great, thank you, Lee. And we have time for one more question. How do I decide which workflows I should use, one-step RT-PCR or two-step RT-PCR?
So one-step RT-PCR is fast, convenient, can be used to minimize the potential contamination introduced by two-step because there's more pipetting steps involved. And that's the advantage using one-step RT-PCR. In this case, the gene-specific primer required, and so the knowledge about the genome or about the sequences is needed. And so this one-step RT-PCR is very extremely popular and necessary for the detection, gene detection world, for diagnosis, quick and sample to results world. So typically one-step RT-PCR is used to detect a virus. But for the research world, in one-step RT-PCR, there's lack of flexibility, and researchers cannot optimize the conditions, everything already added in one tube, and there's limited power for them to optimize their assays. So two-step RT-PCR system will be really typically used by the researchers in the lab, like sequencing projects or any new method developed. To typically use two methods. Two methods, you could use random primers and oligo-DT to basically synthesize the cDNA across the board, and then pick the gene-specific primers to amplify for the second step. Yeah, hopefully I have answered your question.
Thank you, Lee. Do you have any final comments for our audience today?
I want to thank you, everyone, to spend time with me and to going over this all the publications, what enzyme can be used to accomplish what purposes. I'm sure this short presentation cannot cover every single aspect of COVID research with this center stage, our daily life, which is profoundly impact all of us. And I want to first of all thank all the researchers and to endeavor in this effort to overcome this pandemic, provide a solution for us, for all of us. And also we are so proud to provide solutions, reagent solutions to help the researchers. We definitely have large selection of different enzymes. We are well known for the high quality DNA polymerase and our reverse transcriptase. So please check out our website to see what enzyme will meet your needs, and we'll be happy to provide any solutions. If you have any questions, we'll be happy to answer. And please contact our sales team for any questions you have.
Thank you again, Lee, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.